Hello and welcome to Neural Development. This will be lecture number 8 in this series on physiological psychology. I'm Dr. Paul Merritt, and today we're going to talk about how the brain develops from initial gestation through the lifespan. We're going to start off talking about maturation of the vertebrate brain, basically from a prenatal perspective. The human central nervous system begins to form when the embryo is approximately 2 weeks old. It starts with a neural tube. The dorsal surface of that thickens, forming a neural tube surrounding a fluid-filled cavity. This fluid-filled cavity is important because that's where the cerebral spinal fluid goes, becoming the spinal cord and the ventricles. The forward end enlarges and differentiates into the hindbrain, midbrain, and forebrain. So you can see here at about 3 weeks, we get development of the hindbrain, which is, of course, the brainstem and cerebellum, then the midbrain, which includes the tectum and the tegmentum, and then the forebrain, which is the cerebral cortex and subcortical structures. By 11 weeks, we start to get further differentiation, and then at birth, we have a pretty fully developed set of hindbrain, midbrain, and forebrain structures. The rest of the neural tube then becomes the spinal cord, and of course, which transmits information to and from the peripheral nervous system. Development of neurons in the brain involves several processes, including proliferation, migration, differentiation, myelination, and synaptogenesis. So I'll talk about these one at a time. Proliferation is essentially the creation of neurons. Migration is getting them to where they belong. Differentiation is when they differentiate into different types of neurons. They become coated with a myelin sheath through the Schwann cells in the peripheral nervous system and the oligodendrocytes in the central nervous system. And then finally, the development of synaptic connections through synaptogenesis occurs. So proliferation is the production of new cells or neurons in the brain, again, primarily occurring early on in life. Early in development, the cells lining the ventricles divide. Some cells become stem cells that then continue to divide and provide additional stem cells. Others remain where they are or become neurons or glia that then migrate to other locations. Migration then is the next process. This is the movement of the newly formed neurons and glia to their eventual locations. Some do not reach their destinations actually until adulthood. It occurs in a variety of directions throughout the brain. And chemicals known as immunoglobulins and chemokines are part of the important guide for this migratory process. So these chemical pathways are actually what neurons follow to migrate to their appropriate locations. So this is an important milestone because this is something that can be directly disrupted through a variety of teratogens like alcohol. Excuse me. Differentiation then is the forming of the axon and dendrites that give the neuron its distinct shape. The axon grows first, either during migration or once it has reached its target, and it is then followed by the development of dendrites. So this is when neurons differentiate into different types of neurons, such as pyramidal cells or Purkinje cells, depending on where they are located. So they differentiate into their different types, depending on their particular function. Myelination then is the next process. This is the process by which glia start to produce that fatty sheath that covers the axons of some neurons. This is, of course, a critically important process for sensory and motor functioning. Without the myelin sheath, we wouldn't have our sensory and motor abilities that we do. And we know that demyelination is a process by which that process can be lost through such diseases as multiple sclerosis. Again, myelin speeds up the transmission of neural impulses. It first occurs in the spinal cord, then the hindbrain, midbrain, and forebrain, and then occurs gradually for decades. So myelination is a process that continues throughout our developmental timeline. The final process in the development of neurons is synaptogenesis, and this is the development of synapses. This is the final stage of neural development in which the formation of synapses between neurons occurs. This is when we get strengthening of connections between neurons and the development of the connections of chemical communication locations from axon to dendrite. So this occurs throughout the life as neurons are constantly forming new connections and discarding old ones. This is how we learn. This is the entire process by which we develop our abilities. It starts to slow significantly later in lifetime. As we start to get older, we start developing fewer synapses, which is why learning and memory start to degrade as we start reaching our old age. More recently, we've discovered that new neurons are developed later in life. Originally, it was believed that no new neurons were formed after early development. It was essentially that we were born with all the neurons we were going to get or soon thereafter. We now know there's a process called neurogenesis, which is the creation of development of new neurons. So we know that neurons are created throughout the lifespan, and there are some disorders which we think are disruptions in neurogenesis. In particular, there's a current belief that disruptions in neurogenesis is associated with depression. And there's a chicken and egg problem whether or not depression disrupts neurogenesis or neurogenesis causes depression. But there seems to be certainly some indication that these two processes are tied together. So our research suggests that we do develop later neurons in life. We have stem cells, which are undifferentiated cells found in the interior of the brain that can generate what we call daughter cells that can then transform into new glia or neuron cells, depending on where they are needed. We know that in the hippocampus, neurogenesis occurs and is an important part of learning and memory. We also know that new olfactory receptors continue to replace dying ones. In fact, we also know that there seems to be some relationship between development of olfactory neurons and hormones such as progesterone, such that women tend to have better olfactory systems, and pregnant women in particular appear to develop particularly acute senses of smell and are able to identify the scent of their infant soon after they are born. So the development of new neurons occurs in other brain regions. So there's a lot of work done in songbirds, which have a steady replacement of new neurons in the singing area of the brain, which tends to be involved with memory. Stem cells we know differentiate into new neurons in the adult hippocampus of mammals, and this facilitates learning. So we are constantly developing new neurons, and so it's an important part of modern neuroscience is trying to understand the nature of this process and its behavioral implications. The lifespan of neurons really depends on both the neuron itself and, of course, the body that it's in. We know certain things can prolong life of neurons and other things can shorten life of neurons. Drug abuse, inhalants, oxygen deprivation, carbon monoxide poisoning, all, of course, can cause cell death. Skin cells are, of course, the newest of all cells in our body. Most are under a year old. Actually, your cornea probably has the newest cells of any surface of your body. Heart cells, on the other hand, tend to be as old as the person. It's one of the reasons why our cardiac muscle starts to wear out as we get older. There's sort of this lore or belief that we only have so many heartbeats, and the more we use them up, the more likely we are to have a heart attack later. The mammalian cerebral cortices form few or new neurons after birth. Subcortical structures seem to be where most of the neurogenesis is occurring. So we develop new neurons, but they certainly die, and we'll talk about this process called apoptosis, by which those neurons undergo cellular death. One of the important processes by which our neural system develops is by pathfinding of axons and the development of synapses. Axons have to travel great distances across the brain and actually through the body to form many of their correct connections. So there's some research by Sperry that shows with newts indicating that axons actually follow a chemical trail to reach their appropriate target. This is one of the ways in which synapses are formed. Growing axons reach their target by following a gradient of chemicals in which they are attracted by some chemicals and repelled by others. This is consistent with what we've talked about previously in the development of synaptic connections, where the strengthening and connection of neurons can actually cause branching, synaptic branching, and the development of new connections from axon to dendrite. When axons initially reach their targets, they form synapses with several cells. The postsynaptic cells will strengthen their connection with some cells and eliminate connections with others. The formation or elimination of these connections depends on the pattern of input from incoming axons. And so it's actually our experiences that develop these synaptic connections. And so connections will be strengthened by connections that we use. Anything we don't use gets weakened. So this is a process by which we learn and develop based on our experiences. So this is an important part of understanding brain development, is our experiences are directly related to synaptic connections. Important in this process are neurotropins. These are chemicals that promote the survival and activity of neurons, like neural growth factor. Axons that are not exposed to neurotropins after making connections undergo what's called apoptosis, which is a pre-programmed mechanism of cell death. Essentially, the cell essentially explodes. That is, it just undergoes the cellular membrane, starts to disintegrate, and the cell dies. So the healthy adult nervous system contains no neurons that failed to make appropriate connections. In fact, cell death is an important part of development of an adult central nervous and peripheral nervous system. So the elimination of massive cell death is part of normal development and maturation. After maturity, the apoptotic mechanisms become more dormant, so we don't lose neurons as rapidly as we do during development. There is some belief that neurogenesis is part of this sort of cellular restructuring. We know the visual cortex is actually thicker in blind people due to a lack of visual stimuli because they cannot effectively prune out ineffective neurons, and so they actually remain when they're not being effectively used. I want to next talk about the vulnerable developing brain. That is, excuse me, early stages of brain development are critical for normal development later in life. A mutation in one gene can lead to many defects, and chemical distortions in the brain during early development can cause significant impairment and developmental problems. This is where we start to get talking about what we call teratogens. And teratogens are substances that can disrupt normal development. In particular, we're interested in substances that might influence brain development. One of the most talked about current teratogens is the Zika virus. Unfortunately, we don't have enough information about how the Zika virus disrupts neural development to talk about it in any intellectual way, aside to say that it really does, and we're starting to try to understand that process. A process we do understand to a great extent is called fetal alcohol syndrome. This is a condition that children are born with if their mother drinks heavily during pregnancy. Now, I want to note right now that the Center for Disease Control guidelines say that pregnant mothers should drink no alcohol during pregnancy, which is a relatively new phenomenon. It used to be doctors thought one drink on occasion at certain points during the development process were okay. Obviously, err on the side of caution. Probably no alcohol is a better choice during pregnancy. Fetal alcohol syndrome is marked by hyperactivity and impulsiveness, difficulty maintaining attention, some varying degrees of mental retardation and developmental disability, some motor problems and heart defects, and facial abnormalities. And the facial abnormalities are part of the clearest part of fetal alcohol syndrome. Is there's a clear facial structure that you can identify. One of the things that happens during fetal alcohol syndrome is a severe amount of cortical thinning. You can see here the red areas are thinner on average in adults whose mothers drank alcohol during pregnancy. We know that the dendrites of children born with fetal alcohol syndrome are shorter with fewer branches, so we're talking about fewer connections. We know that exposure to alcohol in the fetus brain suppresses glutamate, which is our primary excitatory neurotransmitter, and enhances the release of GABA. Now remember, if a neuron is not excited enough, it will undergo apoptosis because it receives less excitation and exposure to neurotropins than usual, and then undergoes that apoptosis. So by inhibiting the neurotransmission through the facilitation of GABA and suppressing excitation by suppressing glutamate, you get reduction in neurofiring, and then as a result, before a child is even born, they have lost a great number of neurons due to apoptosis. So the cortex starts to differentiate throughout development. Neurons in different parts of the brain differ from one another in their shape and chemical components. Immature neurons transplanted to a developing part of the cortex can develop properties of the new location, but neurons that are transplanted at a later stage of development develop some new properties, but will retain some old properties from where they were. So as we start to differentiate, if cells are moving later in development, they're maintaining some of their previous properties. So there is essentially a developmental timeline and some critical periods that develop because of this, because they have to arrive at their appropriate location and develop appropriately at a certain point in development. The brain has some ability to reorganize itself in response to experience. Axons and dendrites, of course, continue to modify their structure in connections throughout our lifetimes. Dendrites are continually growing new spines, and the gain and loss of spines indicates new connections, which is related to learning. So as the dendritic spines increase and decrease, this is a measure of increases in learning and development of new neural connections. Rats raised in an enriched environment have been shown to develop a thicker cortex, increased dendritic branching, and improved learning. So environment is directly associated with differentiation in the cortex. There's measurable expansion of neurons has been shown in humans as a function of physical activity. So physical activity is a really important part of healthy neural development and seems to stave off neural loss later in life. As old neurons die by apoptosis and new ones form to take their place, there is improved learning and memory. So it's important to understand that apoptosis, that is loss of old neurons, and formation of new neurons and formation of new connections, is part of learning and memory. So this is a particularly important part of understanding how our memories are formed, is through the development of new neurons and new connections. Important to understand our brains are really adaptive, particularly the younger we are. So there are a number of things that we have seen in some special populations. So for example, people who are congenitally blind can improve their attention to touch and sound based on their experiences. Touch information ends up being activated in the occipital cortex area, which is usually devoted to vision alone. But in congenitally blind people, that touch information can take over the occipital cortex area, which is why individuals who are congenitally blind are able to read Braille so quickly and accurately, because there is so much cortex power associated with processing their fingertips to accomplish that reading of Braille. So the occipital lobe, normally dedicated to processing visual information, adapts to also process tactile and verbal information. People who are deaf from an early age also become a little bit more responsive to touch and vision, again because those areas associated with auditory processing become part of what would have been the auditory cortex in those people who are born congenitally deaf. So there is some clear indication that our brain starts to adapt and takes over areas that normally might be reserved for some other function, such as hearing or vision. I'm going to take a little side note on brain adaptation and talk a little bit about musical training. There's a really interesting set of studies talking about how musical training might affect brain development. There are structural MRI studies that reveal the following. The temporal lobe of professional musicians in the right hemisphere tends to be about 30% larger than non-musicians. This seems to indicate that the right hemisphere is probably responsible or somehow involved in the production of music by professional musicians. This thicker gray matter in the part of the brain responsible for hand control and vision of professional keyboard players makes perfect sense. And this suggests that practicing a skill reorganizes the brain to maximize performance of that skill. So this is a clear example of how a skill such as music can take over a part of the brain to change both perception and motor planning. I'm quite sure the same is true of professional athletes. There is some really interesting research that has been done on London cab drivers. London's a particularly difficult city to navigate. And what they have been able to show is changes in cortex, and particularly in the hippocampus, of cab drivers as they learn their way around the city. So they looked at new cab drivers and then came back and looked at them later and found that they actually saw structural changes in the brain because of that experience. So our experiences really do dramatically alter our brain, and our brain is quite adaptable. Now there can be a downside to this. You can end up with what's called focal hand dystonia, or what's often called musician's cramp. And this refers to a condition where reorganization of the brain goes too far. So this oftentimes affects violin players or string musicians. They become clumsy, fatigue easily, and make involuntary movements. And what's happening is the somatosensory cortex is trying to adapt to the use of the dramatic increase in use of these particular parts, so primarily the fingers, and so they start to grow. The problem is that they then start overlapping with the finger next door, so that is they're right next to the next finger. So as a result, instead of just your index finger, you oftentimes will move two fingers instead of one, because there isn't sufficient distinction between the two. They used to treat musician's cramp with hand surgery, which was the complete wrong approach because it had nothing to do with their hands and everything to do with their somatosensory cortex. So the new treatment now has to do with developing the independence of those fingers from one another. And so there are specific treatments available that will actually train the fingers to, again, be more independent and to differentiate those areas of the cortex. So a few other issues in brain development across sort of the decades. Adolescents tend to be more impulsive than adults, which can lead to increased risky behavior, drinking, driving, etc. The problem with adolescents is they haven't developed impulse control yet, primarily because of development of the frontal lobes. And so adolescent period and the teenage years are associated with a lot of risk-taking, because they tend to discount the future and they also tend to discount the risk. And so it's really important that we keep that in mind as we start thinking about how to keep adolescents safe, and also as we start thinking about things like the criminal justice system. So this is an important part of understanding the psychology of development and how that intersects with things like public policy. So adolescents are not equally impulsive in all situations. There are lots of things that can affect their decision making, their peers, the amount of time they have to make decisions. That is it on the fly? Does it need to be done right away? All of these things can affect their decision. The important part of this is that the prefrontal cortex of adolescence is relatively inactive in some situations, and this may or may not be the cause of impulsivity. We know that people become less impulsive the more developed they are. This is one of the reasons why that we no longer allow the death penalty for anyone under the age of 18, because the Supreme Court acknowledged that the frontal cortex is not fully developed, and as a result, adolescents and teenagers cannot be held to the same legal standards as adults because their brains are not adults. So it's a really important part of how neuroscience has affected public policy. Final note, and we'll talk more about this later on in the term, some neurons start to lose their synapses, and the remaining synapses change more slowly than before in response to experiences. This is part of our aging experience. As we get into each functional part of the brain, we'll start to look at how aging affects each of these functions. So brain structures begin to lose volume. We start seeing that research tends to underestimate older people, and that is, people tend to vary in respect to their intellectual decline. In general, older people have a greater base of knowledge and experience, and many find ways to compensate for their losses. And so it really is individual. And we're talking about normal aging here, not any sort of dementia or Alzheimer's. All right, well, that's a brief introduction to neural development. We'll be talking about neuroplasticity in our next lecture.